So I'll launch right into it. I brought way too much material, but I'd really much rather that you interrupt me and ask questions. And I can't actually even see um, the bar with the chat window, so I'm counting on Jeffrey and Liang to shout out when people ask questions. But please do. And I'll happy to drop material to make it in time to have a Q&A session at the end as well. So I'll get through what I can get through, but let's try to make sure that we're engaged here. So I'm actually going to talk at the beginning a little bit about what's the background of the crisis of variant interpretation. It'll be familiar to many of you, but it's worth reviewing. And then I'll talk about a computational exercise, which is, I think, what prompted Liang to reach out to me about a talk. And then I'm going to spend a bunch of time on experimental work in the group. So my group has sort of transitioned over the years from mostly computational to mostly experimental now, but this is a sort of a mixture talk of both worlds. And so the computational piece and then experimental piece. And I'll just set it up here with the crisis that over time we're, of course, sequencing more and more genetic diagnostic panels and increasingly exomes and even genomes. And we're finding lots of pathogenic variants, of course, and that's ramping up, but we're finding variants of uncertain significance, or the dreaded VUS, at a much higher, at a much greater frequency. And so there's some, as and you may know the sort of ACMG classification system, where this is supposed to be like 90% probability of being pathogenic, 95%, and similar on the other wings. And mostly things are falling in this category, especially for missense variants. And that's where my focus is going to be entirely today. And so most VUSs on the ClinVar database of clinical variants are most of the variants of uncertain significance are missense variants, and most of the missense variants are VUSs. So this is the focus. And it's worth noting that 90% of the VUSs are extremely rare. And as we observe more and more VUSs, this is going to keep trending higher and higher. Because we've been sort of looking at more at common variants, but as new novel variants arise, they're typically going to be extremely rare. So why does that matter? Well, so you can associate a variant with being bad by statistics. You can say, it differs in allele frequency between cases and controls. That's the classic sort of GWAS approach. And it's really underpowered for rare variants. And although you can detect rare variants, that's usually at the common end of rare. So for the extremely rare variants of less than, when you get down to 1 in a million, which is what 90% of them are now, there's really not much power to learn something from GWAS. And similarly, you can do family segregation analysis, but that means collecting pedigrees and medical information on a vast scale. And maybe we're going to get there in a systematic way, but the sort of one at a time recruiting families approach is going to continue to be very small scale and resource intensive. And similarly, to identify de novo variants, it means sequencing the parents as well. And that gives you a greater hint that it's pathogenic, but it's, again, small scale and resource intensive. And then there's functional assays that I'll get to in the latter part of the talk, which is to actually measure the functional effects, usually in some cell-based assay. And so we'll go there. But right now, the functional evidence that I will talk about is only about 1% of disease genes have one of these variant effect maps that I'll talk about. So right now, it's not there yet for most genes. And so we're left with computational prediction. And the scale is immense. And so basically, there's a back of the envelope calculation. You can see our review here for that. I actually didn't invent this idea, but we put it first in print with Doug Fowler's permission, and he heard it from somebody else. So, I won't take credit, but I think first in print with this back of the envelope calculation that says that if we had sequenced everybody on the planet today and looked at all the variants, that we would see every single nucleotide change that is compatible with life an average of 50 times. So that means that we have to actually know about all the variants ultimately. And so let's get organized and think about how to do that. And so one way is computation, and that's pretty good because you can do all the calculations ahead of time for every possible variant. and then you're ready. When someone comes into the clinic for the first time with a variant, you've already calculated a score for that variant, and you can think about what it means for pathogenicity. So what are some challenges in the computational approaches? So there's many computational approaches to predict missense and other variants, but I'll focus on missense. And we've put our latest entry into the field here with this AJHG paper that just came out that I'll say more about. But there are many of them, so why do it? Why do it again? And so I'll just sort of say there are four sort of major aspects of a supervised computational predictor, and there's the training data you put in terms of what variants do you use as your training and your test set. And there's features of those variants. What information are you going to use about those variants to help you predict whether they're pathogenic or not? There's how you judge your performance, and then there's what algorithms you use. And so I'll touch a little bit on each of these. And so one of the areas of challenges is choosing a training set. And the issue here is that there are many mislabeled examples. So you can go broad and use as much data as you possibly can, which is good. You want big training sets. For example, HGMD is an enormous training set of information about pathogenic variants, but they say in descriptions of HGMD that they do not filter claims of variant pathogenicity. And so their disease-causing set has hundreds of variants that are probably benign based on how frequent based on their allele frequency. And there are almost certainly other false positives for pathogenic variants from this and other sets. The other issue is false negatives, particularly amongst benign variants, because it's hard to say that a variant is benign. And people often grab sort of random variants from human populations from NOMAD. They aren't totally random because they're made up from case control studies, but even if you use the control subset, there are going to be pathogenic variants mixed in there, especially those causing late onset recessive disorders, that there are actually pathogenic variants in the population. But it's actually a very useful proxy to use just random variants from NOMAD as benign, because they're biased that way. So another issue is choosing training sets that are non-representative. So one thing that people have often done is chosen benign variants from the common variant set. So they're predominantly from common because you can be more confident that it's benign when it's common. And that's great. You can be more confident in them, but they're actually not representative, it turns out. And this is a figure from the Revel paper, Ioannidis, who's now at Berkeley, actually. And they showed that actually the challenge of separating pathogenic and so-called neutral variants gets harder when the neutral variants are rare. So that suggests that the common benign variants, the sort of neutral variants, if you will, the common variants are not representative of the rare benign variants. And so that means that going where the data is highest quality is actually going where the data is maybe less representative of the real challenge, because the real challenge is you have a rare variant in a patient, usually rare variant in a patient, and you'd like to know whether that rare variant is pathogenic or benign. And if your algorithm is, unbeknownst to you, predicting whether something is common or rare, that's not very helpful. Okay, other pitfalls. So people sometimes take as their training set of benign variants that are benign, but they're benign not because they don't damage their gene, but because they are in non-disease causing genes. And so that's not very helpful. And so don't do that. And then the other is using features that either directly use gene identity or serve as sort of proxies, subtle proxies for gene identity. So if you give your machine learning method clues about what gene it's looking at, then it actually can exploit that to sort of, in a funny way, peek at the biases that are baked into the training set and test sets. So if we have lots and lots of variants in BRCA1 because we've looked harder for BRCA1. Of course, there are lots of pathogenic variants in BRCA1, but if we have lots because we've looked harder, and then you build that into your test and training sets, you'll look good in cross-validation, but it actually will fail to generalize to new variants in other genes that haven't been as popular up till now. So that's another caveat. To add to the list, there's things called meta predictors that take as inputs the outputs of other predictors. So you get a bunch of these prediction algorithms, and then you combine them sort of in a combination of expert approaches, and that can often give really good performance. But you got to be careful, because if in your test set are variants that were used either to train your meta predictor or more subtly used to train the input predictors to your meta predictor, then you can be maybe even unbeknownst to you inflating your performance artificially. And I'm actually not going to say too much about performance measures, but we favor the precision versus recall curve methods because they're more aligned with what users think about. So if I get a score this high, how likely is it to be right? And and then what fraction of pathogenic variants can I pick up at this score? Jeff?
Yeah, Fritz, sorry. I wanted to jump in just to help prompt others to ask their questions and things, but I have a broad question. This is probably quite naive, but all throughout the introduction here that you're very beautifully explaining to us, there's a focus on single variants. Is there much work that's trying to extend that to cases where it's pairs of variants that are ultimately pathogenic? That, you know, one variant alone itself is not, but it's actually the combination of two, or perhaps the converse. I recognize that that becomes a combinatorial problem that's monstrous.
Super interesting.
Much that's been done on that. If you have thoughts on whether that is a tackleable problem, or if perhaps you think actually more often than not, the effects of individual variants are additive in a way that you really can't treat them as individual units.
All right, so the challenge of interpreting someone's genome is so hard that we've broken it off into smaller pieces, and the sort of the ACMG guidelines for variant interpretation are all about calling a variant pathogenic or not pathogenic, and that doesn't even think about other variation at the other allele, at the same allele or the other allele, or not to mention genes variants at other loci entirely. So you raise a hugely important point, and I've restricted our focus here to predicting which variants are pathogenic, and that's defined to mean variants that could be pathogenic in the right person at the right time in the right genetic and environmental context. And I'm not going to touch that really at all, except in the MTHFR story a little bit. But it's hugely important, and we need to go beyond predicting whether this variant could be disease causing to actually incorporating that in a disease risk model for an individual. And people are doing that, and there's polygenic risk scores, and there's individual variants, and there's lifestyle choices and family history, and lots of things that can go into that, but it's not the focus of this talk. But it's a hugely important point, and we need to solve it. But I'm focusing on this really narrow problem of, is this variant potentially bad or not?
Makes perfect sense. Thanks, Fritz, for a beautiful explanation.
Okay, thank you. Okay, so next, this is our entry into the race of computational predictors. And it's actually kind of funny. It is a race in the sense that there's a competition happening right now, and the deadline is tomorrow or the next day of the Critical Assessment of Genome Interpretation Challenge, and we're competing in that. And right now we're at the top of the leaderboard, but only because we figured out how to cheat. And we're also doing pretty well with our non-cheater program. But we're hoping to be the best amongst cheaters and non-cheaters in this competition. But anyway, I'll tell you about the past performance evaluations, and then there'll be papers coming on how we do in these challenges. But it seems to be good, and I'll try to convince you. I'll sort of give the punchline first, which is how well does it work, and then hopefully that motivates you to stick through some of the gory details in how it works after you see that it actually does work. And so here is a precision versus recall curve. And again, you could choose where to call it pathogenic at any given score threshold, and where you set that threshold determines the precision, the fraction of time that you're right in calling it pathogenic, versus your recall, what fraction of pathogenic variants do you pick up at that threshold. And so in green is our Verity ER optimized for extremely rare variants, and red is Verity R optimized for rare variants, and blue is the next best one in terms of, here we like the measure of what is your recall at a threshold of 90% precision, sort of assuming that people want good precision or it's not worth doing. So at a reasonably high precision level, what's your recall? And we're at 47% and 40% recall at 90% precision, and the next best one is 37% recall at 90% precision. And if you want the whole forest of curves from all these other methods that we've looked at, the other one was simplified just to show you the next best. But here they all are if you want to go look in gory detail in the paper. And this, I should have said, is rare de novo variants from neurodevelopmental diseases that were used to assess. And these were, by definition, they're de novo, and so they're extremely rare. And so it makes sense that the one we've optimized for extremely rare does better than the one for rare. And here we then broadened it out to basically everything in ClinVar in a cross-validation framework where we trained on 9/10 of it and then predicted on the held out fraction. And I should say that within the training set there, we're doing another layer of cross-validation to do hyperparameter optimization within the training set, but it's all very clean that we haven't looked at the test set. And here Verity R wins, and that actually makes sense because a lot of the ClinVar variants are rare, not extremely rare. And next is Revel, and next is Eigen. But if you want to again see the whole forest of curves, Verity R came out on top in terms of recall, and the rest came behind. But it's worth saying that this looks subtle, these curves look close together, but actually at this high precision threshold, we're actually adding 13% higher recall. And so it's not a totally insubstantial difference, but it gives you the sense that there's a lot of people playing in this game and they're getting better over time, and we're making incremental advances. And maybe I'll talk about how using the experimental data, we could get to more quantum leaps, but let me get to how it works. So why did we do better? And I can't answer that exactly, but except to say how we did what we did and why we did what we did to address some of the challenges I raised before. And so one is that we used a method called gradient boosted trees, and I'm not going to get into the method. It's a classic machine learning method, and nobody would predict that using this would make the difference because it's just a good machine learning method that if people know decision trees, it's sort of like a forest of decision trees. But it does something called boosting, which is it makes it a first tree as a predictor, and then it tries to predict the errors of the first tree using a second tree, and so on, and keeps on trying to make the fit better and better to the training data. But what we did that was different is we wanted to navigate this trade-off between having a lot of data. So we could either have a lot of data of uncertain representativity or quality, or we could have a little data that's very high quality. And so we wanted to think about how do we, get the best of both worlds. And so what was cool about gradient boosted trees is you could tinker with the objective function to give more weight to some variants than others when you estimate performance. So it's not necessarily weighting them more as inputs, except that implicitly by saying that we're going to value performance and try to have the next tree fix the errors, work harder to fix the errors of the more important variants. So we're putting weights on variants. And so we had various sources of, I'll call them proxy pathogenic or proxy benign because they aren't all annotated benign. And particularly, one example is in Nomad. We took Nomad variants as proxy benign, even though there are many contaminants there. They aren't all benign. And then we used weights, for example, based on allele frequency. So the higher the allele frequency, the more likely they are to be benign, but the less representative they are. But we basically used a weighting scheme that in a sort of smooth way weighted variants according to allele frequency. And similarly, from ClinVar, we can weight variants according to how high quality the annotation is, but according to the review stars that ClinVar gives the annotations. And so anyway, we're trading off representativity and accuracy by adding weights. And how do we optimize the weights? How do we choose these weights? And the idea is we have a core set that's small, but high accuracy and high representativity. And we optimize weights to optimize performance in cross-validation on this core set of variants. And that's all. That optimization is all within the training loop. So it's cross-validation within the training set, and then we can go to a clean test set and ask, how well did that work to optimize the weights to perform well on the core set? We also included some new variant effect mapping data sets that I'll talk more about. That may have helped a little. It helped numerically, but not significantly. So I think we're going to need more data to have that make a bigger difference, or we're going to have to be smarter about how we make use of it. So, and so that already began to touch on the issue of features. What information about variants do we use? And we use many of the classics that many other methods have used, sort of the BLOSSOM matrix of which amino acids are more substitutable by others in conservation and structure-related properties. We added a few new properties related to protein interactions, sort of what's the surface exposure that's buried by an interaction partner. And again, numerically it helped, although it wasn't significant, so we're not sure how much it helped. But I think the most important thing we did was take out features. So we actually excluded features that we thought could either directly or indirectly identify which gene you're looking at for that variant. We also excluded input features. It is a meta predictor in that we use the outputs of other predictors, but we excluded supervised predictors, those that had peaked at ClinVar variant annotations. We only took those that were unsupervised, for example, based on conservation and substitution matrices. So this is, you can just close your eyes for a minute. This is too busy, I'm sorry. But basically, we took missense variants from 4,000 human disease proteins, and the variant sets we used depended, of course, on the source we got it from and whether that's likely to be pathogenic or benign based on where we got it from, and then how rare is the variant, because we wanted to optimize for rare variants. And then we put weights. And to first approximation, the core set from ClinVar got 100% weight. And then these sort of add-on sets of less certain quality, or representativity got weights that depended on various quality informative properties. And I'll say more about that. But of course, if you gave a different parameter to every variant for a weight, that's way too many parameters. That's crazy. So we had, for each quality informative property, we had a very simple logistic weighting function. So only a 3-parameter function to just sort of decide how to smoothly weight variants according to this property. And I should say our threshold for Verity R was 0.5% minor allele frequency and 1 in a million for Verity extremely rare. Okay, so these are some of the various informative properties we used. And then again, we optimized weights for performance on the core set. And this is to say that we had a total of 58 hyperparameters to optimize. And again, that was in cross-validation inside the training loop to do that hyperparameter optimization. And you could say that's a lot of hyperparameters, and it is. I don't think it's enough to learn to memorize all the variants we had at inputs. But in any case, we always have to worry about overfitting. So we had this scheme for tuning hyperparameters, ordered our hyperparameter solution. So we're sampling the space of hyperparameters and doing Bayesian optimization to choose the next trial. But we order our solutions by how well they perform on the training set. And overfitting is defined by doing really well on getting better and better on the training set, but not getting better on the held out set. And so we sort of ordered by performance on the training set, and you can see where the performance starts to take off on the training set, but at some point the performance starts to go down in the validation set. This is the cross-validation held out loop within the training set, not the test set, which we haven't peaked at still. So we sort of stop when performance starts to go down in the validation set. So that's early stopping, and we avoid some of the overfitting that happened later. And so just to summarize all this, I already gave you the punchline that it works really well. But the key idea is we kind of make use of a larger volume of lower quality training examples by weighting them, and we choose the weights to optimize performance on a representative and well annotated core set of rare missense variants. And we avoided a lot of pitfalls that I've gone through. And right now we're outperforming all other methods that we've evaluated. And there's actually a follow-up analysis. It's not as well powered, but it's looking at UK Biobank, which essentially nobody is trained on the rare variants in UK Biobank, these methods. And so it's a nice clean set, and you can go look at the bioRxiv paper if you like, but we're tied statistically with Revel in that analysis, which isn't super well powered, but we're numerically better than everybody else, but statistically tied with Revel in that. Okay, so this is mostly credit to Joe Wu, who's just graduated. He was a PhD student in my group, and I'm looking for a job if anybody wants to hire a really talented machine learning guy, but you better move fast. and then a few other people helped in that, especially Jochen, Roja, and Kevin. Okay, that's part one of acknowledgements. I'm going to leap now. So if you want to throw in questions, now's a good time on the computational side, or I'll just dive right into the experimental stuff.
Questions from anyone? Drop them in the chat or the Q&A, and I can unmute you if you have one.
Okay, great. Just interrupt if you see Q&A. Thanks. Okay, so next on to the experiments. So here I want to sort of talk about reactive versus proactive approaches to interpreting variants. So the reactive approach is you see a patient with a phenotype, you sequence the relevant genes that you suspect, or maybe do exome sequencing if it's really confusing. And then mostly there isn't any functional evidence to go on. And if it's extremely rare, there's not really family linkage or population-based evidence. And then you go straight to an interpretation for this patient because you have to, give an answer to the genetic diagnostic that was ordered. And mostly that's going to be a VUS if it's a missense variant. Or you can take a little time and do a functional assay. And mostly that's a research project that's done months or years later. And it doesn't really help the first patient, but it might help the next patient to come along with that variant. And then there's a paper just out on AJHG from Ambry Genetics and folks in Seattle that says that actually you can reduce the number of VUSs down by half with a good functional assay. And so that can really help, but it's usually not there for the first person to come along, or even maybe the next or the next to come along with a variant. So there's the idea then of testing variants experimentally ahead of time and test all of them. So here's 20 amino acids and all the amino acid positions of a protein. And here yellow is the wild type amino acid, so methionine, serine, glycine, etc. But then there are 19 substitutions, and if you just make them all and test them all in say a cell-based assay, you could actually have the experimental data for everything ahead of time and then be proactive so that when somebody comes into the clinic with a new variant never before seen on the planet, you've already done the experiment and can look up the score, and you hopefully get a better clinical interpretation. That's the idea. And by the way, that works for computation. So if computation starts winning, we don't have to do these experiments. But right now it's not. So I'll just give a shout out. We didn't invent variant effect mapping. And so I was inspired by a talk from Stan Fields, and we're now collaborating with Stan and Doug and Leah Starita and Jay Shandur and others in this NIH SEGS Center for Tech Development for this. And so it's really exciting to work with a good team, but I'll represent mostly work from my lab, but acknowledge that there's a huge community out there that's growing in this field. Our take has been, and again, not the first, this is a classic sort of genetics approach, but we've really liked doing the variant effect mapping with complementation assays, and where possible in yeast. And I'll try to justify that for people who think that's nuts. But the idea is that you have a human gene with a yeast cousin where you have a phenotype in yeast when you knock the gene out. And then if the human gene rescues that, and it's a big if, it varies from sort of, depending on who you ask and context, maybe 25 to 50% of these orthology relationships are actually complementable rescue relationships where the human gene rescues. And then if the human gene rescues the yeast phenotype, now you have an assay and you can put a variant in and say, does it still rescue? If so, we guess it's neutral or tolerated. And if it fails to rescue, our guess is that it's damaging. So you could say that's nuts because it's a billion-year diverged model organism. On the other hand, the human gene is doing something right because it actually rescues the yeast phenotype. So we wondered, though, reasonable people can disagree about whether this is a good idea. So we empirically took a panel of 21 genes for which we had these yeast complementation assays, and we made about 140 variants, roughly half pathogenic and half not annotated as pathogenic. And we asked, how well does it work? And compared to polyphen-2, and it's very similar results for the other computational methods we tried at the time, at a high precision threshold, you can roughly triple the number of pathogenic variants you can pick up using the yeast-based assay. So it's not perfect, you don't get them all, but it's way better than the computational approaches. And so we've done most of what we've published is in yeast, although I'll talk about one project if there's time in human cells. So the basic idea is make lots of mutants, and we do this in an oligo-directed mutagenesis way where we mix a pool of oligos with an NNK or NNN degeneracy in the middle and make lots of codon replacements. And we tune that to have an average of 1 hit per gene. And then we move this into a vector system where we can en masse move the library either into a yeast expression vector or a human integration vector. And then we have a little population, sort of lab evolution experiment where if you've got a yeast strain addicted to the function of this human protein, then you evolve it in the lab and you watch the allele frequencies by sequencing. Here, this is a fax picture, but the first example I'll tell you is actually growth, where we're just measuring allele frequency before and after the selection by Illumina sequencing. Then you can sort of infer from the change how much the allele frequencies are dropping out, whether it's functionally damaging or not. And then you get a map. So the big picture is we've done maps for 14 proteins, and I'm going to say more about MTHFR and another one, LDLR, that's just unpublished and emerging in human cell-based assays. And this is an overview of projects in the lab. So these are cases where we've completed a map, the underlined ones are published, and these are projects that are at various stages of the pipeline. So if you see something in there that looks cool, give me a shout. I really like to collaborate with people who are experts or have access to clinical cohorts where we could judge the quality of our maps. And I'm just doing a time check. Link, do I have 10 minutes or so?
Yeah, definitely.
Okay, cool. Thanks, Jeff. All right, so next I want to talk about 2 case examples, MTHFR, which does not stand for what you think it does, and the LDL receptor, LDLR. And then if there's time, think about sort of the big picture in variant effect mapping. So this is the first map. I like this example because it's the first map where we vary environment and, per Jeffrey's earlier question, genetic background, and see how the landscape of variant effects changes in different genetic backgrounds. This is probably a simpler example than Jeffrey had in mind. But in any case, we want to look at variant effects and how those change in environment and genetic background. I was inspired by a talk from Jasper Ryan, who'd worked on this, and he developed, and Warren Kruger had developed MTHFR and CBS assays. That we have a paper on that I won't talk about. Anyway, MTHFR is methylene tetrahydrofolate reductase, and it's in the 1-carbon metabolism pathway. And it's early onset, the severe cases are early onset homocystinuria, and sort of a little bit of history of the disease from first discovery in the early 70s, quickly recognized as an enzyme defect, really quickly recognized that folic acid, increased dietary folic acid could help, and some patients, but not others. And that turned out to be interesting. It's not, folate is interchangeable with folic acid and drives production of the precursors. So some of this may be mass action, but it's more complicated than that because it seems that high folate levels also help retain the cofactor FAD. So it's a more complicated story. But anyway, on to gene cloning and this yeast complementation assay developed by others. And particularly Jasper developed a version of this that is also where you can control externally the folate available in the cell. And so you can look at the effects of folate levels or folinate levels on the function. So
Fritz, we just got a question from Michael Rogers. Michael, I'm going to see if I can find you in the list and allow you to talk. If you want to ask your own question, if you prefer not, I'm happy to ask it as well. So let's see if this works.
I think it works. Can you hear me?
Yes, great.
Yeah.
All right. So going back to where you were talking about your biological selection assays, I guess I would imagine that that those complementation assays, especially in yeast, are going to do a better job of identifying mutants that are not compatible with life, as opposed to disease-causing mutants. Is that what you observe, or do you actually see significant discrimination of disease-causing alleles?
Yeah, it seems like good question. It seems like the answer is going to be different for different genes and assays, and the threshold at which a function is bad enough to cause disease is not the same as the threshold at which we can detect effects. So although we can get these functional scores from the assay, to know what that means in terms of predicting pathogenicity, of course, you need controls of known pathogenic and benign or proxy benign, and then see where the scores thresholds separate. But there are definitely examples where we're quite clear that there's a functional effect, but it doesn't cause disease. And actually on this slide is the A222V variant in MTHFR, which is 10% of us are homozygotes for this variant. And although there's been a lot of sort of newspaper articles and controversy about this variant, it's probably not that bad. Like if pregnant women get enough folate in their diet, there's no evidence really that it's damaging, except as long as pregnant women get enough folate in their diet. And so this is a great example where we clearly see a functional impact in yeast, but it's probably not of great disease importance.
So you're saying it can actually go the other way as well?
Yeah, so there's, I'll also give examples of places where the yeast assay doesn't work so well. Like there's some aspects of MTHFR's function where the assay doesn't work so well. And there are aspects where it works well, and then sometimes maybe too well, that it can pick up subtler functional effects that are not important enough to cause disease.
Okay, great, thank you.
Sure. Okay, so well, actually, and I'll say the first maps we did were in SUMO1 and UBE2I, the SUMO conjugase, which are super important biologically. Many of you know the SUMO tag, but actually not at all important from a disease perspective, at least for missense variants. There are no known Mendelian missense variants, mostly because I think if you screw up either of those two proteins, you're dead, and so there's no disease. It's basically embryonic lethal. Okay, so back to A222V, it's very frequent. Half of us carry it, 10% of us are homozygotes. It does have a functional impact. It has about 65% activity of the wild type. And other than this neural tube defect risk, which is remediable by having the recommended sort of folate diet, so it doesn't seem to be a major disease risk, just except for that worry about diet for pregnant women. But to investigate this question, like, is it really so okay to have this variant? We want to investigate further and also think about environment. So this is the first gene where we did multiple maps in different folinate levels or different environments, in this case, folinate levels. And then we did 4 folinate levels and then the same 4 again, but this time every clone, in addition to picking up the mutation we're interrogating, also every clone carries this A222V and cis. And so we're sort of starting to think about genetic background effects. And at a high level, these look like replicates, but I'll say a few things. So anyway, the maps let us get at deleteriousness, folinate response, and the effect of this background variant. And yes, indeed, we did pick up this variant as being damaging in the assay and worse at low folate and not so bad at high folate. So that made sense with the literature. But interestingly, even though there were not that many variants that depended on folinate level, this is sort of how things shift in terms of at somewhat arbitrary color scheme by how damaging the variant was, but there's not much of a shift in it as a function of folate level. But in the A222V background, there are very many more variants that are now becoming dependent on folate. And so there's this complicated interplay of genetic background and environment that begins to hint that we've got a lot of work ahead of us in terms of the incomplete penetrance sort of question that Jeffrey raised earlier. How do we actually predict individual disease risk? But then I'll just jump to the folate sort of, you can sort of interpolate the maps between the maps and say, how does the landscape change at different folate environments? And this is our favorite example of a particular tryptophan that's super important at low folate, but not at all at high folate. And there's a whole story I don't have time to give justice to, but it looks like folate in the active site helps keep the FAD cofactor around. And in the absence of folate, there's this loop, especially this tryptophan at the tip, that flips in and we think helps stabilize the cofactor. And this is flopping all over in the breeze when you change it from a aromatic to a non-aromatic. So we think we've got an explanation for at least one case of a highly environment dependent variant effect. I guess the money shot here is the everything in color is something derived from one of our experimental maps. So there are various ways to cut it, but they're all better than all the computational methods that we tried, all previous to Verity. So I should go back and check each of these to see Verity starting to do better. I doubt it still, but Verity was just a bit better than the others. Anyway, the experimental map is still the way to go to separate pathogenic from benign for MTHFR in the catalytic domain. But actually, the assays seem to be less useful. And you can almost see that in the maps, that the map is sort of faded in this regulatory domain. There aren't too many disease-causing variants there either, so it's not a huge loss. But in any case, we seem to do better. Oh, I don't have the regulatory domain. In any case, everybody does worse computational and at our map, and we do better than computation still, but worse than the catalytic domain. Okay, I'm going to jump to big picture. I'll just say this is our first map in human cell-based assays, the LDL receptor, and I'll give the punchline and say we just have the first couple of regions of it, not the whole thing. And many of you know of LDL as the bad cholesterol, and this is sort of the N-terminal set of 7 LA repeats that bind LDL. The receptor binds LDL and helps pull it in. And the first early conclusions from this map are of these 7 repeats, the first 2 don't seem to matter at all, and the 6th doesn't matter at all. And this part was known, and I think this is new, that this 6th repeat doesn't matter so much, and that these are the important ones. But in the interest of time, I'm going to jump ahead to the sort of big picture outlook. I just suffice to say that this all works in human cell-based assays as well. It just takes longer to set up and longer to do, and there are more points of failure, but it absolutely works. And when it all clicks, you get a good map. And this is to say we've done it by 2 different methods, bar-seq and tile-seq, and we get almost the same map. Okay. A big picture, I guess I just wanted to say a few things that, we're starting to get organized as a community. There's a database called MaveDB to share this kind of data. We now have visualization tools that you can grab this kind of data from MaveDB and make visualizations of it. There are a few tools out there now for that. The count of publications, the curation needs to be updated, but the number of publications is still growing with these variant effect maps. And the number of variants per publication, so this on the y-axis is here is number of variants, is growing with publications. So people are tackling bigger and bigger proteins with this approach. And what else to say? I guess of the roughly 4,000 human disease-associated genes, maybe 5 to 10% will have a yeast assay, and we like yeast when we can get it, but I think it's fair to say we're all going to get better at human cell-based assays at taking those larger scale. And almost 80% of human disease genes have some sort of phenotype in a large-scale CRISPR screen. And so actually the prospects are quite good that for a large fraction of human disease genes, there will be an assay. And by the way, LDLR wasn't on this list. And so if you just sort of read the literature and scratch the surface on interesting genes, you can come up with cell-based assays often. So I think we're going to get close to 100% of human disease genes that will have a cell-based assay. And you could say that's crazy because that, there are these sort of organismal tissue level diseases, but you know what? MTHFR is an enzyme that works in cells. And we had a yeast-based assay that worked pretty well to separate pathogenic from benign. But the phenotypes of MTHFR are of homocystinuria are intellectual disability and Marfan syndrome and other organism level phenotypes that yeast don't model, but you can still make a cell-based assay. I'll just say we have tools that we've published for prioritizing genes. This is our sort of difficulty adjusted impact score that looks at which genes we should all start to hit first. And so initially we went where the assays were good, but now we're actually have a check-2 map in preparation. VHL is in progress in my lab, and STK11 and others a little farther down the list are in progress here. And I helped co-found this Atlas of Variant Effects Alliance, and I hope you'll all, if you're interested, look into it and join. But right now we've got 200 members in 10 countries. And the idea is to sort of get together and facilitate this as an international community to make these maps. And I don't think it's a crazy vision to say at least for every protein and for sort of proximal promoter regions, et cetera, and ends of introns. And anyway, we could probably assay within the next decade even, I think it's not crazy, to annotate the functional regions of the human genome in a sort of a first draft of these maps. Although the devil's in the details and modeling environment and genetic background, et cetera. We also have something called MAVE Registry, which is to actually have people not only do open science in terms of preprints, but actually advertise what they're working on as they're working on it. So you can know who's working on a map for what gene. And so there are now 65 research teams and 150 projects registered here. And so if you're curious to see who's working on your favorite gene, go check. If you want to nominate a gene and say you guys should be working on it, you can nominate a gene there. So please check that out. And so I'll just stop there and we can do some Q&A and just thank lots of people in my lab and outside of the lab for doing all this hard work, and Jochen and Song and Nishka on the MTHFR map, and Atina and Daniel on the LDLR map that I didn't get much time to talk about. I acknowledge Joe Wu on the earlier computational stuff, Kevin Kwong and Jochen on a lot of the tool building. And I'll just, lots of people to thank, but they're here on the slide, and the slides will be on the web, I guess. So thanks. Maybe I'll stop sharing and open it up.
Thank you, Fritz. Really very beautiful talk. Questions? If you wouldn't mind, please post them in the chat or the Q&A, and I can again try and give you the permission to ask your question yourself. Looks like we have one from Shira. You've got your hand up.
Hi, thank you. That was great. I was just wondering, you mentioned clinical cohorts being relevant when looking at this. Like, what specific kinds of clinical cohorts would be most interesting or useful for the genes that you've mapped so far?
I mean, each one is its own story, but I guess for LDLR, we were in pretty good shape with the UK Biobank because they've collected LDL and other, triglyceride and other lipid information from a vast majority of the participants. So that's great. And we can start thinking about how well our map stacks up against computational methods, just using that cohort data and trying to predict in individuals what their LDL levels are from their genotypes. But right now we're on the hunt for STK11, which is a somatic cancer gene, but also as a germline variant causes something called Putz-Jaeger's syndrome. And so if there were case control cohorts there where we could say, not only because I want to go towards what Jeffrey brought up, which is to actually think about individuals and not just say, stop at the classification of pathogenic versus benign. And so I'm really interested if quantitatively, if our function, the extent to which you've lost function in the gene relates to age of onset or severity of symptoms or type of symptoms, or if you have hits in this domain versus that domain, how does that change the disease? So I'm super interested in that. And so for that, you need a really carefully phenotyped cohort where someone's really thought about this. So that turned out to be true for CBS, the cystathionine beta synthase we did a map for, and our scores correlated with age of onset and severity. And it was true for age of onset for MTHFR. We're having a hard time finding any quantitative effect with HMBS map, which is done but not published. But that's a super, it's low penetrance disease and highly variable and dependent on environment. So maybe that makes sense. Yeah, I don't know. If you have a cohort that sounds interesting, let me know.
What's the number of patients that would be relevant for something like this? Because UK Biobank, it's huge.
Yeah, no. But I guess whatever you can imagine correlating a variant score with a phenotype. So you just need to have enough different variants in the cohort, right? If it's 100 people, we're thinking about alpha aminotrypsin is one gene we're thinking of, and there are 2 recurrent variants that cause disease, but you see a lot of those 2 variants and not a lot of the others. And so it's not about number of patients so much as sort of number of variants. And so if you had even 20 different variants and phenotypes for those, I think you could, measure a correlation between scores and phenotypes.
Okay, maybe I have one question. Maybe it's a naive question. So Dr. Roth, can you talk a little bit more about the feature selection? I think the machine learning method you use, you basically use the weighted variance, and I think you also consider about the gene conservation, protein-protein interaction. So is that mainly based on your domain knowledge, or basically you try each features one by one and see whether that contribute to the model or not?
So there are two places where features get used, and we thought more about one and less about the other. So in terms of what features you throw into the gradient boosted trees, we sort of threw a lot of things in there, and we only sort of, we threw out things that we thought could be sort of hidden proxies for what the gene is that you're looking at. But we were willing to try lots of features as long as we didn't think it was sort of peeking at gene identity. We threw lots of features in there that may or may not have been helpful, because the gradient boosted trees essentially do feature selection for you. And so we didn't worry about putting too much in unless we thought it was going to cheat in a way that was unhelpful. And but then the other place we used features was to decide how to weight variance, as sort of deciding what would be a quality informative feature. And that was sort of domain knowledge to say what to try. And then it's described in the paper, we had this sort of primitive approach to decide whether it was quality informative, where we had a slightly longer list of features that we whittled down if it didn't look like it was going to be promising. But mostly it was just, kind of makes sense that variants would be somehow more valuable if they had a higher quality annotation, like ClinVar review stars, and allele frequency was pretty obvious once Revel pointed out that there were that higher allele frequencies weren't representative of lower allele frequency variants. I don't hope that helps.
Thank you. Yeah.
Okay. 2 minutes early, everybody gets 2 extra minutes for email, or we could do more questions, hang out.
Any last questions? All right, well, it looks like there are no additional questions, Fritz. So once again, thank you so much for joining us today.
Thanks for the invitation. It's fun. Thanks for your questions.
Thank you, guys.
All right, have a great
Bye.
afternoon, everyone.
Okay, ciao.